In this video, we will discuss about one of the last figure of this CAPPSID system, where they developed a system that in which the capsid can be controlled using this TEV protease. So why did they decided to make such kind of modifications? Is because the virus sometimes some of the virus has the properties to spread systematically, and it can cause some side effect. Because of that, the structural proteins of the virus has been mutated or is been dysfunctional, and this limits the replications of these viral capabilities. So in order to improve its replication capabilities, they decided to modify their capsid system in a way that can replicate the virus even in the place where bacteria is not able to infect the cells. So for that, what they did is they modified the structural proteins cleavage site you with this TEV protease site. And so what they did is in the virus that they are using, they generally produce big one big amino acid, and that cleaves at different sites. So these cleavage sites are being replaced by this TEV sensitive cleavage site, where TEV can cleave these sites to make the structural proteins that can thereby start functioning, or that can thereby replicates the viral RNA. So this is their plan. So they check their hypothesis in a case where the cells are not transduced with TEV protease and the cells are transduced with TEV protease. So for the wild type, where they do not make any changes in the uh, this cleavage site, they can see same number of signals or these GFP signals. But in the case where they cleaved the protease site with these TEV sites, they are not able to see any signals in the case where TEV protease is not there. But when the TEV protease is there, they are able to see the signals. And they did their studies at different cleavage site, and they can see that they did their studies by keeping this TEV at different sites. And what they are able to find out is that when they keep the TEV at this first site, they are able to make a systematic virus replications compared to the other case. Although we can see that the TEVs when it's kept between VP2 and VP3 are able to reproduce faster, but this is this they thinks that is uncontrollable at some time, because the modifications that occurs at this site because of the RNA prone polymerase that leads the virus applications even in the case where TEV is not there. That's why they choose the second site, that is TEV when kept at this between before BP4 is the best candidate for the further studies. So this is how their next plan is, that they keep the TEV before this BP4 or cleavage protein 1. And what will happen is that during the translation, this TEV is translated, and then it will cleaves off different uh, structural protein so that they can start functioning towards these viral replications. And when they check its results, and they are able to see that when the TEVs are only present, or when they are able to see that when TEVs are present, and or when this capsid virus is present along with this TEV protease is transfected, they are able to see a transfection or percentage transfection is increased. And it is comparable to the case where wild type capsid system is used, where protease where a cleavage site is not modified with this TEV cleavage site. And when the cleavage site is modified with TEV cleavage site but there is no TEV, they are not able to see any transfection. And these things they have quantified in the panel D, and the fluorescence images are shown in this panel E, where we can see that when the protease is not there, in that case they are not able to see any significant signals. But when the protease is TEV protease is also there, they are able to see significant signals. Next, they wanted to check next they wanted to check, or they optimize this TEV based on the literatures, that some modification has been done in the TEV in a way that will prevent the non-specific replications of the virus even in the absence of TEV. So they replaced some of the amino acids that they replaced. Not able to recall its name, you can refer to the reference. So once they have made this mutated TEV, what they are able to see is that in the case where only TEV is there, there is no TEV protease, and the sites are modified with this TEV cleavage site, they are not able to see any signals. And when the TEV protease is there and the sites are modified with TEV cleavage site, they are able to see some transfection or infections. However, when the TEVs is been modified, they are able to see some increased infection rate that can be seen here, and that is comparable to the case where wild type virus is been used without any modification of the cleavage site with TEV sensitive TEV detectable cleavage site. So they finally use this modified TEV for the in vivo studies, and in the in vivo studies they are able to find out that the results that they're seeing from uh, the SVA, where cleavage SVA or capsid system, where cleavage sites are being modified with the TEV detectable cleavage sites, and also in the case where recombinant TEV is used, or the modified TEV used with the TEV protease. So in both the cases, they are not able to see any significant increase in the infectious particles, and these signals are also comparable to the case where they use wild type SVA without any modification in the cleavage site with TEV. So in the case there is not, in the in vivo studies, there is not much significant increase with this mutated TEV. Next, they wanted to check the tumor luminescence in the case where this TEV is injected, and the wild type system is there, and what they are able to see is that, what they are able to see is that when this TEV protease is not there, and in the case where TEV protease is there, so when the TEV protease is not there, they are able to see a decrease in the signals, indicating that the virus is not replicating further. Whereas when they also introduce the TEV protease along with this recombinant, or along with the protease site, it's been modified with the TEV detectable sites. In that case, they are able to see luminous signals for a longer duration of time, and this even at 14 days, the difference is statistically significant, indicating that the modified TEV system is they modified capsid with the cleavage sites modified with the TEV detectable cleavage sites is controllable by the TEV protease, and which can in in such case the applications can be stopped or reduced uh, using by reducing the TEV protease in the system. So that is what they have finally uh, proposed, that their capsid system is the TEV dependent capsid system that can be used for this that can be used for diagnosis, and it has a high it is a high viral infection it has a high viral infection capacity, and which is also controlled by this TEV protease. I hope you understood their TEV dependent control of capsid system to some extent. If you need to understand more about their experiments or their system in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.